and we have started on 19th, and today is 26th. In between, we had a class on 22nd. Anybody who has joined late today itself, so welcoming them again here. So, and I hope you are kind of getting glued into the whole event of things here, which is happening, which are going to happen in the next 3 months of the course. And also, I would request you to to visit the educational site. I hope many of you have made the profile out there. Now let us go and find out what we kind of did on the last day, 22nd of July 2019. We kind of touched upon something which is called big data bioinformatics. Under that, there were topics like knowledge driven versus data driven discovery, and also discussing some examples about on these particular aspect. And we have kind of went through briefly on, touched upon on the applications of bioinformatics in agriculture, neuroscience, cancer biology, immunology, defense, and healthcare. So most of these topics that are kind of covered in July 22nd lecture are elaborately covered in our introduction to bioinformatics course that is there in this link. Many of you, I have seen that kind of have gone through it. Please complete them as you go on, because this is going to be very important for you guys to really understand what is happening and what kind of things do you expect to learn here. What kind of things do you would you really learn at the end of this entire exercise, at the end of this entire course. So on the right side of your screen, probably all of you can see my screen. So I guess so. If not, I think you can write in the chat. Our team, which is there supporting this entire process, would be happy to help you out to make your things, make your problem get solved. So this is about, so it says bioinformatics for clinical audience. This was a poster which was displayed by Donna Slonim of Tufts University as ISMB conference in 22nd of July 2013. So since it has been quite an amount of time when people realized that bioinformatics is a very important part of any sort of biological experiment, number one, and also at the same time, it's important in the clinics as well. So what is the problem that she is trying to address out here? She addresses the problem, a medical doctor who says that I wish I learned how to understand big data. Look at the time, 2013. And at that point of time, this lady is giving you a slide which says, I wish I would have learned how to understand big data. So the time has come to really understand how the big data is making the changes in the biology. So what is the gap out here? The gap out here that, okay, you have a medical training. At the same time, you are working in you are working also in certain amount of molecular diagnostic, understanding molecular diagnostics features from the clinical findings that you are having. Trying to relate the molecular features of the cell to the clinical findings is a gap. There's a gap in between. The doctor is very adept in trying to look at the clinical perspectives or the clinical, the clinical features that is prevalent in a group of patients or in a single patient. But why does this happen? I mean, what is the reason for these kind of things happening? Now there's a gap for that. What is the need? The need is to understand the genetics of it, and it's also needed to have a systemic thinking, and also at the same time, we need to learn maths, and within maths we need to learn probability and statistics. So we have a big data, we have a huge amount of data available nowadays from high throughput experiments. It was also available. It has started being available in the year of 2013. And now we need the genetics and we need a systemic thinking to really understand what the data tells me. And at the same time, we need to understand mathematics in parlance of probability and statistics. Now imagine a doctor who is coming from the biology domain. He or she need to have that ability and need to have that pick up those skills which are necessary. What happens in US? In US, undergraduate schools have liberal arts, and instead of liberal arts, this lady says, we need to teach them computational biology and statistics at this level in a very basic parlance. That's what we are trying to do here. People who are kind of joining in this course, many of you, most of you are undergrad students or just entered into your postgrad days. So that is why it is important for you to take up courses like this so that you can understand the various facets which are necessary, which are absolutely essential when it comes to biological research and biological jobs that are coming up in the market. So this lady says that in the medical school itself, after, in US, the system is totally different. So let's not go into that detail. But when you are learning the level of course you are in there in your life right now, you have to kind of understand the computational biology stuff and bits and pieces of mathematics and statistics. Maybe many of you are learning as a part of your curriculum as biostatistics. So that is important for you. And what do she also kind of tells you that, okay, in Tufts University, they have statistics and computational biology. Please do not go into this detail. What I'm trying to focus out here is that there is a need to understand genetics, systemic thinking, maths, and probability, not even for a doctor. So in 2013, it was for a doctor. But right now, it is kind of, you have to learn it. You have to understand it. You have to be, you have to get skilled into it. And definitely there are organizations like Pine Biotech, which will help you to become enabled and which will help you to become skilled in this particular domain. So, there are things which, and obviously, what happens at some point of time, this is a very important concept in any of the, in the world right now. Bring your own data. So let's say many of you, I mean, I remember in the last class, somebody called Orijit, who is a assistant professor at a university in West Bengal. He said that I have some transcriptomics data. Would I be able to analyze it? Yes, of course you can. But the point is that first you have to learn what analysis you need to do. You have to, our whole motto is basically to enable you people so that you can analyze your own data. So somebody who is a scientist, he is already having a data from some high throughput experiments. The students who are actually doing the course, they can download the data from some data sources, which we will going to be showing you in in next few classes. So you possibly many of you have gone through this introduction to bioinformatics lecture, and I have seen one or two have completed it also. And I would request you all, I request all of you to be, to actually go into this introduction to bioinformatics thing and figure it out that whatever I have told in the July 22nd lecture is actually present out here. And you have to do it. To become an expert, you have to start from the scratch. And even if there are expert people out here who has been there in biology for a pretty long time, I would request all of you, it is never it's always good to go back to your basics and do it, refresh it. Because if your basics is all right, then only you will be able to go forward and really do, novel things. You can do fantastic things in the future. So progress, we are in the third lecture today, and let's look at the progress right now. So this is what is the graph which is present out here. So we can see the progress. For example, we have seen that it has started on July 19. Today is July 26th. I have picked up this graph in the morning. So let us go to try to figure out what do we have here. So hopefully all of you can see my screen. Will take a while. Okay, here we are. Okay, great. So, wow, I love to see this peak. Wow, lovely. So, yeah, it is important to go into this thing. Ayushman has done a lot of education. He has logged on to the server. Fantastic. This is good. This is really encouraging. Okay, so we are halfway through the July 26th. We already have starting catching up. So this is the gray area that you see is basically the server usage. So Chito Broto, your Amity University assistant professor, who will be taking the class in the next lecture, he is using a lot of server to show you certain examples. I will be giving you a small overview of things of the server today. Shubhrojit is using a lot of server. Okay, we have new leaders. We have Madhuvendra, we have Shreyoshi, we have Devangana, and Satyanaran is already there. So many of you have joined. Joydeep Pal, Arnob, okay, fine. Arnob, yes, he's a new entrant in our Amity program. So yes, please go on. Please do your stuff. Please, go on to the introduction to bioinformatics. And after this lecture, today's lecture, we are kind of, we are now kind of ready to delve deeper into the world of bioinformatics. So in the next 2 days, it would be very good. It would be very nice, really, if you can complete the entire introduction to bioinformatics. Okay, and that would really help you to catch up in the next few classes. Okay, great. So yes, so these two reviews we have received. Shreyoshi, you have completed the introduction to bioinformatics course. Thank you for that. It was a very enlightening course, as she says. I hope it was really enlightening to you and you have understood many things. And we also have, it's very encouraging, for us to really see this sort of review, because these people like this, they have kind of given some time into this course, and beyond your existing curriculum, beyond your existing duties that are there every day at the university and also at your home. It is very important. I mean, I'm going to tell you one thing, because if you say that I don't have time to go to the introduction to bioinformatics, that's like not really done, because all of us have time. Okay, it's not like that we don't have time. We have time, but the point is that we do not really use it judiciously. We use a lot of time to really do nothing, basically. So in that nothingness, if you just put in this thing, it is very interesting. And of course, if you put some extra effort into this particular process, it will definitely going to reap you benefits in the future. So SNAG, I would request you to write your full name the way that Shreyoshi has written, and also put a picture of yourself so that we know you. We kind of know each other. So as you can, as I was pointing out in the last lecture, that we are trying to build a forum here of interested people, of initiated people, of people who are really they want to learn something, and of course they want to exchange thoughts, they want to exchange something which is important, and also have a very fruitful discussion among themselves in the domain of bioinformatics and beyond. Okay, going forward. So this is the today is the lecture on high throughput data. So it's all started with DNA, deoxyribonucleic acid, which was and it contains 3 billion base pairs. So, and it contains, it's a double helix model, and one strand goes from 3' to the 5' end, another strand goes from the 5' to the 3' end. So it's anti-parallel strand. They are kind of intertwined between themselves as a staircase, and they have alternate, they have base pairs, C, cytosine, guanine, thymine, and adenine, and they are packed into the nucleus, in such a way so that it says that if you really extend that entire chromosome, uncoil that chromosome, it can cover quite a distance. I mean, if I am not wrong, or if I have not forgotten, maybe it would have covered the distance from the, if you really uncoil it, the distance between Earth and the Sun can be covered twice if you really uncoil the entire length of the chromosome. So you can understand how much packing is done. So there are very interesting, things that which says higher order DNA structure. Higher order DNA structure really understands the packing of the DNA into the small nucleus, and how do they really uncoil, and while the DNA really wants to, when a cell replicates or when a cell divides accordingly. So from the DNA, a very important concept emanates, which says central dogma of biology. What is the central dogma of biology? It starts with the DNA, okay, and it starts with the DNA, and which produces RNA, ribonucleic acid. So deoxynucleic deoxyribonucleic acid produces ribonucleic acid, and thereafter from RNA protein is being produced. So this triangle is basically called. So you would say, okay, this dotted line, does it really mean some amount of direct production of protein from the DNA itself? Yes, somewhat. In bacteria, only in bacteria, directly from DNA to protein is being produced. So this completes the triangle. So from the path from DNA to the RNA is called transcription, and from the path from RNA to protein is called translation. DNA also produces its own copy by a process which is called replication. So as you can see, this figure has been divided into two parts. On the left, which is the gray part, is the nucleus, and there is a part on the right which is the cytoplasm. So this is the this is within the cell. There are distinctly two separate places where the the transcription is happens, and thereafter, after the transcription from the RNA to the protein, translation happens. So many of you who have done your post-graduation, we are doing your post-graduation or doing your BTech in biotechnology, or maybe in your third year of your graduation days, maybe you are already aware of it. So, but as I was telling you, that bioinformatics starts with the basics of biology as a whole. So you have to kind of touch upon those things when it comes to biological interpretation of the results that are being produced by the pipeline that you are going to use. These sort of ideas will definitely be important for you to come up with a biological insight. So from the RNA, yes, RNA also replicates. Yes, they do replicate only in virus. So many of the virus have RNA as their starting material. RNA viruses, which we say, and RNA viruses also produces reverse transcription. And in viruses and some of the cells, some enzymes, reverse transcriptase enzymes are used within the cell, are kind of present in the cell, which produces DNA from the RNA itself. Okay, there's a very interesting thing. From the RNA, there is something called microRNA, miRNA, which is a part of epigenetics as well. So which actually controls the DNA replication and also the transcriptions. So in the last class, probably I have told you that, only 0.5% or maybe 5%, 0.5% of the entire length of the, entire length of the genome is actually transcribed into RNA. What happens to the other part of the DNA? Earlier, it was called junk DNA. Actually, this is not junk. These are the DNA. This is that part of the DNA which produces a lot of RNA, which are not translated into protein, but actually they modulate the functions of the DNA and also the functions of many of the RNAs. Also modulates other functions of many of the proteins as well. These are sometimes called microRNA. They're sometimes called LNC RNA, long non-coding RNA. They're sometimes called they're called sorry. So they are called miRNA, LNCRNA, non-coding RNA, ncRNA, and so on and so forth. So only the mRNA, which is produced from the DNA, which is called the messenger RNA, actually translates into the protein counterpart. Okay. So on the right, so replication, another explanation of the entire event that is taking place. DNA polymerase is the chief enzyme which is important when it comes to the replication, where DNA is produced from another DNA. When it comes to transcription, production of DNA, for production of RNA from DNA, an enzyme called RNA polymerase, RNA pol is, so RNA polymerase is the whole enzyme family. So there are a lot of types of RNA polymerase which are coming into play, producing the RNA. And also after that, from the RNA to the protein, there are this is a very serious thing. Anything which goes wrong here, I mean, in any of the states, as you can see, if anything goes wrong, it can produce, myriad results. It can produce many type of variations and cause different types of things. Okay, so we have 23 pair of chromosome, right? So the up to the 22nd pair, they are called autosomes, and the 23rd pair are called allosomes or sex chromosomes. So our sex chromosomes are two types. Basically, in females, they have identical copies of X, and in males, we have one X and one Y. So as you can see that on the right, this is a electron micrograph of X and Y chromosome that is present in humans. And so any disease that are being produced, so on the left, this figure is basically the karyotype. This is also an electron micrograph, but this is a karyotype. Karyotype means that what are the number of how does the chromosome actually look? Okay, so as you can see so when it comes to cell division, a little bit of detail on this. So when actually the fertilization takes place, haploid cells are being produced. The haploid cells connect with each other to form the diploid cells, and the diploid cells actually forms the embryo, and the embryonic development takes place to form into a zygote. And it's for after the zygote develops into an embryo and into an organism as well. So this picture is important because offspring gets an equal amount of autosomes 1 to 22 from the father, and equal share of each of the the sex chromosome from either of the parents. So as you can understand, if you really calculate, if you really produce a graph, that that is why a girl child gets an X from the mother and Y from the father, and a boy child will get X from the mother. The girl child will get X from the mother and also the X from the mother, or X from the father. But a boy child will get X from the mother and Y from the father. So that is why the sex differentiation takes place. Going on, so if you really go deeper into the structure of the DNA, so as I was telling you that there are antiparallel strands from 5' to 3', and from so the length, the orientation of the DNA is from it's always from 5' to 3', but it is always reversely placed, right? So in one strand, if this is what you called the nucleotide. So a phosphate bond and the phosphate, the nucleotide, and also the nucleotide base is actually called a nucleotide, and therefore the bonding of between C and G, cytosine and guanine, takes place by three bond. It's a triple bond, and bonding between adenine and thymine takes place by a double bond. So on the right, so there are two classes of nitrogenous base in the DNA. One is called adenine and guanine. One is adenine and guanine. This is called purine. And another one is thymine and cytosine. They are called pyrimidine. So the significant difference between purine and pyrimidine. Yes, Ognivesh, you have some question to ask me? Okay, thank you. So purine have one furan ring and one benzene ring. But on the contrary, a pyrimidine does not have any, only the benzene ring and nothing else. Okay, so how does it happen really? So this is the kind of really trying to look at the central dogma as in the with a microscope. So this is the entire length of the DNA, which is antiparallel supercoiled structure. So how does it happen? A gene, any gene 1 or gene 2 or gene 3, let's say gene 3 is getting transcribed into RNA. So this is the RNA that is being produced. This is the length of the DNA. So from 3' to the 5' end. And when from the DNA transcription happens to mRNA, it says that the only difference between between the DNA and the RNA is a base which is called uracil. So instead of thymine in the DNA, in the DNA, instead of adenine, instead of thymine in the DNA, in the RNA itself, there is something called uracil base. Okay, so typically it says any sort of signal that has been produced are produced in triplet. They are called codon or triplet codon. And from mRNA, when the protein is getting translated, they are getting translated into various types of amino acids. Let's say TRP is tryptophan, PAG is phenylalanine, GLY is glycine, SER is serine. Now this chart on the right gives you an idea about, since there are 3 positions of a triplet code, you can clearly find out that which of the DNA code, which triplet code produces what type of protein at the end of the day. And if you really calculate, if you really try to go back and kind of create a combination of the type of occurrence which might occur, which might happen in a particular human body, how many proteins can you really say? I mean, if you really take the first position, what are the various options of the first position it has? If you go to the second positions, how many options do you have? So combinations of these 3 letters can produce a huge number of options of amino acids at the end of the day in the body. Okay, so we understood few things about the DNA, and we know that the DNA is replicating by itself. The DNA is transcribing into RNA. The RNA is translated into protein at the same time. So as I was telling you, if anything goes wrong at this level or at this level, will produce a lot of things, lot of difficulties. So this is a cartoon which has been shared by me by one of my colleagues. She is a clinical bioinformatician, and so there might be some proteins which are important to prevent cancer. So when it is at the normal stage, when it is under a normal state of reaction or under a normal state of function, it would prevent cancer. Some variation occurs, some mutation occurs. What is a mutation? Mutation are some abnormal changes which changes the structure, which changes the function, which changes the composition of the DNA at the subcellular level. When the mutation happens, it is changed. It is losing its function. It does not really care. It says, okay, I don't care. I mean, whether cancer is being produced or not, I don't really care. Okay, so this is the result of the mutation or the kind of things that might happen when any cellular mechanism that I showed you, if the central dogma goes wrong. As you can see, When anything goes wrong at the DNA level on the left, what kind of things can happen? It can lead to abnormal methyl transferases. Okay, these are some of the disease. These are some of the abnormal conditions that are produced inside the body. It can produce abnormal methyl binding factors, which are otherwise known as Rett syndrome. It can produce abnormal parental imprinting, which can lead to Prader-Willi, Angelman, Beckenwith-Whitman, Silver-Russell disorders. So these are all genetic disorders, as you can see. So anything which goes wrong at the DNA level produces these sort of myriad happenings within the body. What happens when what happens when DNA goes wrong or hypermethylation occurs? It produces cancer. It produces autoimmunity. It produces neurological disorders, allergy, cardiovascular disease, and aging, and so on. Going forward, according to the central dogma, the molecule that comes into attention is RNA. What happens when anything goes wrong at RNA? Variations in RNA-dependent DNA methylation. As I was telling you, that many of the RNA which are being produced as as control mechanisms, which takes part in the control mechanism of the DNA replication or DNA transcription, if they go wrong, they can, if they go wrong, they can produce different results. So what does it say? Variations in RNA-dependent DNA methylation. So methylation is a very important factor in the level of the central dogma. So if something goes wrong at the methylation level, it can produce regulation of de novo methylation, control of retrotransposable elements. So retrotransposomes, they are called. So these are something which controls the signal mechanisms of the DNA and also controls the produces those or regulates the effects of how the DNA replication or the DNA transcription will take place. What happens when variations in what happens when variations in deposition of RNA goes wrong? It produces hybrid dysgenesis. It produces variations in the pigmentation phenotypes, produces transgeneration cardiac hypertrophy, and also abnormal RNA editing, which is also very important because during the central dogma, one thing I have really missed out is called post-transcriptional modifications. If so, the RNA is reorganized among themselves, it produces something called embryonic lethality, neurological disorders, longevity, and obviously cancer. So anything else, this part is known as epigenetics actually. Okay, we do not know exactly how does the epigenome of the body really works. We are trying to understand it because most of the DNA which is wound inside on the histone proteins of our body actually have that have hidden signals which might or might not be produced, and also if it is methylated, it will not be produced. If it is non-methylated, it will be produced. So methylation of DNA is a very important factor to really understand how the DNA functions. Okay, anything which goes wrong at the protein level? Self-replication of sporadic or acquired prions. Prion is a disease. Prion is a very, it's almost a very unsolved disease that happens among the Caucasian population a lot. What happens when things go wrong at this point? It produces transmissible spongiform encephalopathies, potentially other neurological disorders. Okay. What happens when something goes wrong at the abnormal chromatin remodeling? It produces something called Chard syndrome. It produces neurological disorders and of course cancer. And abnormal histone modifications, this is the epigenetic control of the cancer. This is something called imprinting disorders are also produced, neurological disorders are also produced. So if you really look at the number of different different events that are occurring due to the improper functions of these molecules, one common word that you can find in all the places like DNA or RNA or protein is cancer. That is why cancer is the most myriad disease, because it can be produced from different sources. That is why, people always are, even if they are trying to look at cancer from a particular perspective, it is always very important to look at it from the perspective, from all the perspectives at all. And that's why we are trying to study cancer from the multi-omics perspective as a whole. Okay, so all these events that are occurring, all these changes that are occurring in the molecules of DNA, RNA, or protein, how do we quantify that? Because we need to quantify, we need to understand how much effect. It's not only about qualitative understanding, it is very important to have a quantifiable understanding of the stuff. It's very important. That's why we come to this point. What is a simple quantification would look like? Counting the reads in aligned to each DNA, okay, in each gene. As you can see, these are all small sequence fragments which are produced from some high throughput experiment, and this length of DNA has been randomly fragmented into these small pieces, and we are trying to align this thing to really find out what are the sequences, what are the exact sequences, whether they do match or not. This particular gene have 8 fragments and this particular gene have 4 fragments. So this is the DNA, this is the gene location that we have, because as you can understand, many of the genes that are being really translated into protein. So eukaryotic chromosome have 2 significant parts. One is called exon, other one is called intron. Exons are the portions which are actually translated into proteins, and the introns are actually snipped away. They are kind of, they're just moved away. So when you really get that mRNA and reverse use some chemicals, or let's say enzymes which are similar to reverse transcriptase, if you use them and get back that part of the DNA, you will not be getting the introns. You will only be getting the exons. Now you want to have to align them. Okay, so this is what it is taking place here. You are trying to align them on the gene and trying to figure out that, okay, what are the exon part of the DNA and what is the intron part of the DNA. So this is a simple quantification. Now think about it, that this length of DNA is let's say 100 nucleotides long. Probably it will be easier, or probably it will be a manageable thing for a human to sit for a week to really align those reads onto the genome. But if the DNA is 1 million nucleotides long, do you really think that a human will be able to do it? We need algorithms, we need machines to do it. And thereby, the importance of machine learning, the importance of all those bioinformatic methods are coming into play. Okay, quantification. When we start with quantification, we come across a method which is called next generation sequencing. What is next generation sequencing? Next generation sequencing is something which says that is a transformative technology that is redefining the landscape of human molecular genetic testing. It enables unprecedented parallelization of sequencing reactions, facilitating highly multiplexed testing paradigms with relatively rapid turnaround time and decreasing costs. So, with 2 important factors. One is relatively rapid turnaround time and decreasing cost. What do you mean by turnaround time? We mean basically that when we are trying to sequence a particular genome to really understand how things have changed within the genome, we need the results very fast. We need to analyze them and we need to come up with interpretations. If you have really, because many of you, I think many of you have done the Introduction to Bioinformatics course, or at least those who have done it have come across certain particular concept out there which is called molecular diagnostics. Now, bioinformatics and next generation sequencing go hand in hand to really understand molecular diagnostics at the point of care. Point of care is nothing but the clinic. Many of the clinics nowadays around in India also are providing you all those services connected to next generation sequencing. Okay, so what happens? A growing number of diagnostics laboratories are embracing next generation sequencing and using it to drive new DNA-based test offering, ranging in size from multi-gene disease-specific panels to entire exomes and the rapidly emerging use of complete genome sequencing. So he is known as the father of sequencing, Frederick Sanger, who started in the year of 1977. So the machine that was kind of developed is called ABI systems in 1995 and 1995. If you really look at the the sequence of events, it starts in 1977, where 700 bases per day the machine was able to sequence. If the technology would have remained at this stage, it would have taken 118,000 years for a human genome sequencing to get completed. So the technology advanced. In 1985, what happened? The first automated sequencer was discovered, where 5,000 bases per day was the people were able to sequence. If the technology would have remained at this level, it would have taken 16,000 years, right? So again, the technology, these are all high. I mean, this is the birth of the high throughput experiments that you see nowadays. So in 1995, where the ability was really, upgraded to the level of 19,000 bases per day, it would have taken 4.4 thousand years to complete the human genome. So since 1999, we have, the technology advanced to such a level where they can really produce 400,000 bases per day. And anyway, if this technology again would have remained the same, would have taken 205 years for the human genome to get completed. So as you can understand, the need for the high throughput technology was imminent. It was absolutely necessary. You cannot go around it. You cannot go without it. And also at the same time, if you go around it, you won't be able to produce that much amount of, you are not being able to, give that thirst for knowledge will be, it will be a setback for the human population, human civilization. Okay, a few minutes back, I was telling you about NGS being used in DNA-based test offering, where multiple disease-specific panels or entire exomes or complete genome sequencing are happening. So basically, these three types of next-generation sequencings are available. They are if you start reading from the right, it is whole genome sequencing, starting with whole exome sequencing, and targeted panel sequencing. So this is the most generic sequencing technology that are used. All genes and all non-coding DNA is being really read, trying to understand that what are the sequences. And obviously, it has a lower coverage. What do you mean by coverage? The coverage is that the accuracy of the data. Somebody was asking me in the last class about veracity. So veracity have a connection with low coverage. So the correctness of the data is very important, because if you do not have the correct type of data, you won't be able to figure out, you won't be able to really understand, derive a meaningful biological insight from the data itself. Coming back to whole exome have intermediate coverage. It's a little better. As you can see, the graph really, this is also a quantification, because as you are really trying to understand or really read the sequences, the quality of the data that is being produced is very important. So it has an intermediate coverage. Going forward, it says the targeted panel sequencing, which are, it encompasses 40 to 400 genes, and it has a very high coverage. It is very accurate at the same time. And one more thing I would like to tell you about Sanger sequencing is that a variety of molecular biology, chemistry, and enzymology-based improvements have brought Sanger's approach to its current state of the art that we kind of saw in this particular thing. Okay. And this is really the advancement of next generation sequencing to a level where nowadays we can get, you know, a number of genomes can be really sequenced in a very short span of time. So this is the process of this particular slide will tell you that how actually the next generation sequencing occurs. I guess all of you. Yes. Anybody? Any questions you have? Want me to give you a pause? Let's look at the chat. Okay. We have so many things. Okay. Okay. What is hybrid dysgenesis? Okay, hybrid dysgenesis is a disease, is a genetically, is a genetic disease. I mean, I think the best place to check out for this type of thing would be to really go to, NCBI or any place and trying to figure it out. Can you please repeat DNA editing, abnormal editing? Okay, so DNA editing or abnormal DNA editing is something that, you know, when anything goes, so DNA, when it reads itself, it's called replication. So something can go wrong. I mean, there are molecules, there are enzymes which really take care of the corrections. You know, if you make a mistake, it really corrects that DNA. But if anything goes wrong in that reading, the mistake remains in the DNA. So either it get transcribed into the RNA, and thereby it can get translated into the protein. So editing of the DNA is something which are they are the places. These are the places where actually all those difficulties happen. Difficulties happen within the DNA molecule. Okay, Shomnath Dotto, what is 370377? I don't get your question. Are you Indian notation? Okay, fine. Thank you, Shubhrojit. Okay, Arup, can you repeat again? What does coverage mean? Okay, Arup, what by coverage we mean that, you know, the read quality. So you will learn something about single end, pair end thing in the next few classes. So you will get a more idea about coverage. You will get a more idea about depth of coverage. Coverage is something like whether you are really being able to take care or really able to understand what exactly signals that are being produced. As I was telling you a few minutes back, a lot of molecular biology, a lot of chemistry has come into this, this reading of the DNA, this next generation sequencing. So there are many chemical principles which are typically used when NGS is being done. So by doing all these experiments together, we are trying to increase the accuracy of our reading. So coverage is nothing but trying to be more accurate about the reading of the DNA or the reading of the signals that or the reading the molecule, to be very specific. Was it okay? I guess so. Okay, so moving on, we don't have much time left. So this is the, if you have the slide already, if you really read it, it is being fragmented into small parts, as I was telling you. And, you know, all the DNA, when they're fragmented, have sticky ends. So there are some adapters which are added to it. Okay, otherwise they will stick to each other. So these adapters are added and they're kind of attached to the DNA to a surface. And thereafter, there are, like, you know, some dideoxynucleotides are added onto the solution, and then they are kind of fixed onto a plate, and thereafter the double-stranded molecules are denatured, and in the part B they are actually being, the DNA is allowed to grow. So that is DNA dependent, RNA dependent DNA polymerase are being used to really grow the exact strand, and we try to understand that how does really DNA replication takes place, and we read the DNA, we get the signals. We use a lot of laser to really understand, and we get signals. So the intensity of the light tells me what kind of nitrogenous base actually is getting added into the DNA. So again, see image of the first chemistry cycle after laser excitation. Capture the image of emitted fluorescence from each cluster on the flow cell. Record the identity of the first base for each cluster. So we get signals like that. So if you go to databases like ArrayExpress or microarray data, this sort of thing are a lot of used out there. So a number of reactions are being done. Sequence read over multiple chemistry cycles. So this is also, the question that somebody asked about the coverage. This is also one more important thing, that if you read a particular portion, since it's a mechanical reading, there might be chances of errors. So we read the portions a number of times. When you read a portion number of times, a large number of fragments are being produced. If you remember the quantification slide that I showed you, the quantification slide has several fragments of a particular DNA. How these fragments are produced? Actually, these fragments are produced from each run of the cycle. Each run of the cycle, in the cycle that you see right here on this out here. So each run produces a fragment. Maybe one of the bases are missing out there. So we align those things on the top of the DNA, trying to understand that how much coverage, how much correct data that I have been able to produce to get the imprint of that DNA, which is unknown to me, the sequence, etc. Okay, these are some of the sequencers available nowadays. So this is, this is something called PacBio. This is this machine are Illumina machines. Illumina machines are used nowadays in a huge, they are kind of very much in use. Probably in West Bengal, there is this NIBMG, National Institute of Biomedical Genomics. Probably you can go there and see the and the Illumina machine, Illumina high-sec machines out there. So, but again, Illumina high-sec machines and all these things are important for short read. They're called short read sequencers. So the length of the fragment would be smaller. And these machines are basically long read sequencers. This is a handheld device nowadays. This is Oxford Nanopore machine. This is also a sequencer, and these are important. And short read sequencers are more accurate. So as you can see, the small amount of DNA, small amount of length of the read will produce more precise results. This is the type of data that are being produced. As you can see, this is the Illumina machine that you see on the left of your screen. So we produce something called FASTA format, and these are somewhat very similar to text files that you can open these files in your notepad or any text editors. But mind it, if the file is, let's say, 1 terabyte, or because it is it contains the data of the entire genome, be it a bacterial genome or be it anything else, it could be very huge. And we attach some quality. This is the most important part, right? So FASTQ format, we say it at FASTQ format. Many of the files that we will be using in our analysis server will be in the FASTQ format only. Okay, so this type of data are being produced from the Illumina high-sec machine. So this is how the pipeline that you're going to use, the server that you are going to use will look like. So I guess many of you, most of you have, all of you have the username and the password of the server. I guess all of you have been able to log on to the server and kind of see this sort of screen. Now let us go to to the server itself and find out how does actually it looks like, how does a sample pipeline looks like, and you can also do it on your own at your end. Okay, let us log on to the server. I'll give few minutes break so that all of you can log on to the server, and then probably look at the demo that I'm going to give you. I will, if you don't have the link, I will paste it right here in the chat. Use your credentials that have been shared to you over email. You should be able to. Shoham says, okay, if a file contains so large like 1 terabyte, then how can we open it? I mean, why do you need to open it? What do you need to see? So there are programs to see the file. So there are software programs. That's why you need to use certain tools. That's why you need to use some certain bioinformatic tools to really see what is there in the file. And that's why we use tools like this that you can see in my screen right now. I mean, it's not like a doc file or a docx file or a pptx file or an Excel file, or let's say your something like that. It contains data. It contains meaningful or maybe unstructured or something like that data. But if you need to open it, you have to use the proper tool. You cannot just go to that particular file, double click it and open it. The difference between you who are kind of aspiring bioinformaticians or anybody who is not being who is a very a user, a simple user, is that you at the end of this exercise should be able to know, should be knowing the fact that, okay, what software, what tool do we really want or do we really need to know to understand or to open a particular file and see it. I mean, you cannot just double click it. That is for sure. Okay, I guess most of you have logged on to the server. So the pipeline that we saw, this is a pipeline which is used for analysis of genomic data. Okay, so if you go by the color coding, so as you can see, there are 1, 2, 3, 4, 5 sections to this analysis. The first thing is pre-processing. Pre-processing is very important for any analysis of data because it removes the artifacts of any of the sequencing experiments that are being done in all these machines that I showed you. So we will learn about pre-processing, the concept of pre-processing, the significance of pre-processing, etc. But what I told you in one word, that is the most important part. What is the second part? The second part is the mapping, the quantification, which I showed you. The third part is variant calling. What is variant calling? Variant calling is basically trying to identify what difference or what exact signal is there in a particular position. What is the fourth point? Differential variant calling. Fifth part is the annotation. Annotation is something which really makes us understand that, okay, whether the results that we have find, we verify that result with something. That's called annotation. What is the sixth part? That part is the post-processing. Post-processing is very important to represent the data in a proper way, in a human readable format. And furthermore, as I would like to say that when Shoham asked the question like whether these files are how can we see inside? So these are basically text files. That definitely there are .txt extension file, SAM or BAM or FASTA or FASTQ. But you need special tools to open those files. It's very important to if you really want to see it, if you want to visualize the file to see whether the data is useful or not or something like that, you need special tools to open them and trying to understand what is there inside. Okay, so yeah, let's go to the pipeline. So this is the pipeline that we ran. Can you see my screen? So maybe, yes. So this is the pipeline, the variant calling pipeline that I'm going to use. Okay, so this is the same image actually, right? So you do a start. So do not worry about uploading or downloading the file. Okay, let me start from the beginning again. So this pipeline needs files to run. But what we have done at our end, we have already uploaded this file in the server. So you don't have to bother about the input thing, about inputting the files out here. Okay, they need some files to run. This needs some, this is a demon. It needs something to eat. It needs something to run. What does it need? It needs files to run. But you don't worry about it. We have already uploaded the necessary file out there. So you just have to run and see. So if you do a start, there's a pop-up window. The pop-up window says what this pipeline is going to do. It says demo pipeline job tutorial. So what this tutorial is telling you? This tutorial is telling you that what is this pipeline made for? Read these lines. These are all educational pop-ups made for you, people like you who want to become, who are trying to become bioinformaticians. It talks about mutational variants. Mutations are variants, blah, 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 whatsoever. If you scroll down, you can see all these things that I showed you in the slide. And it explains pre-processing, removing technical duplicates and adapter trimming, mapping on a reference genome, aligning reads to the reference sequence and annotating the sequence in a GTF file is available. Variant calling, what is a variant calling? Determining which positions have changed nucleotide from the reference genome and differentiating consistent change from the random, and so on. So go on reading it. You will understand everything. So, but again, you have to read, otherwise you won't be able to understand. So click OK. Now you can see a few highlighted places, right? You can see PCR clean, you can see bow tie 2D. Now all these boxes are basically algorithms. Sliced FASTQ, clean primer, sliced genome, PCR clean. These 4 algorithms fall under the class of data preprocessing. If you click on each block, you can see what is this block made for. So see, this is the data preprocessing block. It says data preprocessing is a technique that is used to convert the raw data into clean data set, blah, blah, blah. Please read it, okay? After, this should be your exercise when we are, till next day when we are starting to do this, we'll be running a lot of pipelines. So each of these set have pop-ups. Read it, why this block is made for. A beautiful picture will be attached. So read it, okay? So let us select. So if you want to select what is the next step, let us select PCR clean. Again, this, if you click one thing, click one particular box, it will tell you what that box is going to do. See, PCR clean is a procedure that removes technical duplicates reads from the FASTQ file. So here, this is the input, FASTQ. This is the input of this particular pipeline. Okay, so click OK. See, this arrow is being built. So the connection, we have already started building the pipeline. We have not started the pipeline. We have started building the pipeline. What is the next immediate step that is being highlighted? Bow tie 2. Now, please remember, this pipeline is an AI-guided pipeline. What do you mean by AI-guided pipeline? This highlighting. So it intuitively highlights you to select the optimal steps, the steps which are necessary. So it is meant for experienced users. It is meant for users like you guys who are just starting to learn bioinformatics in your graduation or your undergrad days. So that is why, that's the beauty of it, okay? Because we don't kind of talk in the air. We kind of give you that thing. See, this is the pipeline. This is the AI/ML pipeline that we have. This is the server that we have given you access to. So bow tie 2D is an algorithm. Click on it. This tells you what bow tie 2D does. Bow tie 2 is a memory efficient algorithm for aligning sequence reads to large reference genomes. It is designed to work with a range of read lengths from 50 up to thousands of characters. So if you really go back to the place, if you can really, try to remind yourself of the slide where those whole exome sequencing and whole genome sequencing and targeted panels are described, think about the numbers that you saw out there. So selection of the algorithms will obviously help you, will be guided by the knowledge you have. If you don't have that knowledge right now, you can read it right here. And there is also associated publication widget. Click on it and you will see the publication which are connected to bowtie 2T algorithm, bowtie 2 algorithm. Okay, see, bowtie 2 is being selected. So we have started, we have done PCR clean, we have bowtie 2. Once we do bowtie 2, the other algorithm that are kind of highlighted right now is Strelka. Very interesting name, Strelka, right? It's a very interesting name. So this again, another educational prop-up about Strelka. It tells you how the Strelka works. And if you go down, you can find a publication. If you can see my screen, you can see a link highlighted out here, right? Okay, so click OK. See, Strelka is being built. So after Strelka, another algorithm, it is automatically highlighted. This is the guidance. This is the AI guidance that we tell you. Select smooth somatic sniper. This educational pop-up will tell you what does a somatic sniper algorithm does. Select it. Okay, we have selected PCR clean, bowtie 2, Strelka, somatic sniper, and then the next immediate block that is being highlighted is end, right? So see, we have done data pre-processing, we have done mapping. So the immediate thing after mapping is differential variant calling. So this is the optimal step. This is the minimal step. You can elaborate it, obviously, but this is the minimal step. So start from the basic. This is the basic variant calling step that we do in our genomics pipeline. Select end. Okay. So after this pipeline runs, you will be able to see the results immediately in your pipeline. You can see the output files in folders by jobs or download the final output. If you select visualization, you can see link to a JBrowse instance where the varied data is displayed. This is your task. So yeah, anybody wants to ask me any question? You want me to repeat? Okay, we have 37 chats. Oh my God.
Could you please explain what a genome, reference genome is to people? Thank you.
Okay, so yeah, reference genome. Arnab Banerjee, what are reference genomes? Apart, you have already answered it. Reference genomes are those genomes which we use to map our reads to. Reference genome depends on the reads you have. If you have reads of humans, we use human genome. So what Apar says, 38, this is the 38th version of it. The first version is, 37th version. So we map it to the reference genome. As I was telling you, we can only sequence the exome part of it. When you are actually sequencing the exome part of it, or we are trying to really understand we are reading the entire genome, we have to map it back. We have to map it back to the source to see whether the mapping has taken place in a proper way or not. That is the reference genome. So reference genome can be human. Reference genome can be, when you are dealing with certain, mutations, not mutations. We want to check. That is one of the purpose. Definitely. Thank you, Mohit, for answering this question. Yeah, definitely. Trying to understand mutation is one part, but there are other purposes too. So, okay, repeat the process of the pipeline. Okay, that's what I was trying to tell you. So
Hello?
if you want to deselect, yes.
To be more specific, I want to ask that what if I am sequencing a completely new organism? What then?
Give me an example.
Suppose I have a new plant sample. So I want to make the a sequence its genome. Then what to do then? What will be the
Then how do you know that genome is not already sequenced?
pardon, sir?
How do you know that genome is not already sequenced?
Because it is not available on any of the public databases.
Okay, so you start with searching about the genome that you want to sequence. So that part of sequencing is known as de novo sequencing. So we start from the scratch. So there are other processes to really understand what are the exact signals that we have got. So that part is called de novo sequencing, when we are trying to understand that, okay, how does your new genome, which you have sequenced completely, which is something very new, can be used. Does that answer your question?
Yes, thank you, sir.
Okay, great. So yeah, repeating the pipeline. So let's say there's a very interesting thing that I also would like to point out. If you want to deselect a particular algorithm, you right click on it. Now that we have kind of given to the end, you cannot do it. So I will reload the server again.
Sir,
Yes, Akash Bhattacharya, please tell me.
sir, after I press the end button, sir, it is showing output files, download pipeline output files, and
Yes,
somsniper.zip, and it
right.
is 71,28,470 bytes.
How many megabytes would that be? Tell me.
Uh,
Yeah, so what is your question? Sorry, please go
sir,
on.
so if we want to see it, will I have to download it and then I have to visualize it?
Exactly.
Yes, sir. Thank you, sir.
No, I mean, okay, visualization is something else. First, run the pipeline.
Yes, sir.
Okay, so this again, this is a demo pipeline that I'm showing you. So you can, what I would suggest you, Akash, is that try to understand running the pipeline is the last of the steps.
Okay,
Try
sir.
to understand what algorithms are being used out here. Read about the paper, read about the educational pop-ups. Then only you will be able to understand that what algorithms produces what inputs are required in a particular algorithm to run. That's the interesting part of the bioinformatics. It's not only about running the pipeline and just getting the results.
Okay,
Getting the results is definitely important, but the point is that it's very important to understand what algorithms are coming into play. So
thank you.
let us keep the request. Thank you, Akash. Thank you very much. Okay, so Shomnath, for you, I mean, for the rest of the people who are also there. So we start. We get an educational pop-up out
Pratim,
there. You are okay.
Pratim, hello.
Yes.
Can you go back and show them how to go into the demo pipeline? Many of them are not able to go inside the demo pipeline.
Okay.
Slowly, just if you can show them once.
Okay, great. So let me ask you, all of you have logged on? Has everybody logged on to the server? If you have logged on, if you go on the right,
Yes,
you
sir.
see, yes, Shomnath.
Yes, sir. We are locked on.
Fine. So if you have logged on, all of you have logged on, then you will be able to see your name out here. For example, I have logged on with my username and password. That's why it shows protein@pine.bio. It will be something else. Your email ID will be shown here. And you will be seeing a screen like this. And as I was telling you, I mean, before I really begin, I should have told you this at the same time. So it starts with NGS data. So this block of algorithms out here on the left, the leftmost panel talks about or helps you to analyze the NGS data. So if you hover around, if you just go here and put your mouse cursor, you don't need to click. You just have to, yes, if you click, you can get all those things. So you will get all these details out here, right? if you just keep your mouse button here, it will tell you what is next generation sequencing. If you click on it, if you left click on it, it will tell you all the details. Okay, it talks about the technology, it talks about the, you know, the pyrosequencing thing. Please go on reading this thing, okay? Before asking questions upfront, please go on, read it. Now you have sufficient, you know, you have sufficient material to read. You have a lot of homework to do. So do your homework and come back with questions. Okay, so this is a very interesting thing we already have. See, there are tutorials. Right? So this is a server related things that you have here. So if you go here, let us start from the beginning. NGS data, you left click on it, you get a pop-up, you click on the tutorial, you can directly click on it, or you can right click and open link in the new tab. Okay, great. So yeah, keep this operation, keep this activity for you as your homework, let's say. Okay, so we are trying to analyze NGS data in relation to the mutational variant or the parallel analysis of mutational variant data. We are trying to analyze that. Click on the, you know, small arrow that you can see. If you can see my cursor, there is a small hand which is there on the down arrow right here. Click on that. You will find something like this, demo variant. If you click on this again, it will collapse. If you click on that, it will, you know, open up. It will say demo. It says variant calling, cell lines variant calling. Click on this, on this demo. You will find this, you know, this elaborate set of boxes, elaborate set of algorithms that are classified into various steps. So they're classified into data pre-processing, mapping post-process, blah, blah, blah. You click start, right? I don't think we have much time today, so I'll quickly go through it again. So the immediate next step that needs to be done with this set of data, which is already there, uploaded by us, is PCR clean. Click on this. Now, if you really want to, let's say you want to go back, you know, you don't want to select PCR clean. What you can do is you can right click on it. Pop-up will again say, are you sure you want to jump to the previous step? If you click OK, it will go back to the start. If you click cancel, it will stay here. Now let us do OK. See, it goes back, right? So PCR clean is the next immediate step. Bow tie 2 is the next immediate step. We click OK. Then the immediate next step, which is highlighted in the pipeline, is Strelka. We again, you know, do this. Then we have another one called somatic sniper. And then we select this thing. The next immediate step is end. Now, if we click end, you cannot go back. End is the step which is something which is embossing. You know, it is the last step. So by clicking end, you cannot go back to the previous step. Up till here, you can always go back. Okay. But when you click end, you cannot just go back. So any one of you want to know about these genomics courses, you are most welcome to go to our edu.t-bio.info and all the genomics courses which are out there, introduction to genomics. But before jumping onto the genomics, please complete, you know, do complete. I mean, please is a, you know, with a long stress, I'm telling you, go to the introduction to bioinformatics and complete it. That will be the, you know, entry point of you into the domain of bioinformatics. That will really help you out. Click OK. The pipeline is complete. See, this pipeline has already been completed by us, so you don't have to worry about it. What you need to understand here is that how a pipeline is being built. So as Akash was telling, we already have a downloaded file. Download this file at your own computer. Try to look around it. It will not definitely, you know, as Shoham was telling, it will not hurt your computer a lot. So there will be text files out there. Try to open it in your text file, in your notepad files, and then ask us questions. Okay, so yes. Going back to the presentation. So this particular things were already there. So this pipeline that I showed you belongs to genomics, and you can get a lot of idea about genomic courses out here. If you go to edu.t-bio.info collections genomics, and this is kind of end of today's lecture. I would go forward to tell you about you know, we have already launched a program called Omics Logic Genomic Data Analysis, which has started on July 23rd. The classes happen online on Tuesdays and Wednesdays, and the classes are being conducted by our international team and also our Indian team as well. The topics that we cover are processing genomic data, identifying DNA variation, analysis of the variants, annotation, and using the machine learning for genomic data analysis, and definitely independent project work that you are also supposed to do at the end of this bioinformatics course at Amity. So it's a 3-month long online program. So it is we have a fantastic clinical bioinformatician in our team who is based out of Greece, and she works in a hospital out there, and she has kind of she's one of the chief instructors of this program. Okay, so sharing with you some of our you know, developments in India. We have tied up with Sister Nivedita University. As you can see, Dr. Mohit Mojumdar sitting out here. The person in the red jacket is actually Professor Anil D. Sahasrabuddhe. He is the chairman of AICTE. So Pine Biotech has joined the industry leaders with like NASCOM ticker plant. So we aim to give you, the kind of collaboration that we are trying to do. We are trying to enable the people of the universities and institutes to give them the right kind of understanding about how the data science and bioinformatics and the biological data marry together, and therefore SNU has collaborated with us. Okay, so we are going to start very soon a pilot program, a program which will be inculcated into their course very soon, and therefore this collaboration is going to be very fruitful for the students, beneficial for the students, and very fruitful for people who are trying to understand bioinformatics or biology from the data science perspective. This is another organization we have collaborated with. They have, I mean, we both have collaborated with each other. BioClues. BioClues is a bioinformatics club for experimenting scientists across India and across the world. We have fantastic scientists. Fantastic. One of the fantastic scientists we have in our in this BioClues organization. We have collaborated together to give sufficient this mentor-mentee relationship to understand bioinformatics from different perspectives. So you don't worry whether you don't know bioinformatics. That's why you are here. So here we give you that enlightenment, that enablement, which will take you forward. So the next lecture would be like on 29th of July, where we will start learning about the transcriptomics part, and actually next generation sequencing in the domain in the parlance of transcriptomics. We'll be talking about pre-processing, and we'll be running a few pipelines, and then mapping the transcriptome and the genome and the combined strategies, and of course quantification of the gene and isoform expression. This lecture will be taken by Dr. Chitrabrata Mal, Assistant Professor of Amity University. Most of you who studied at Amity know him very well. So this will be on July 29th. So on July 30th, we have a workshop at Amity University, which is going to take place on big data in biology, or whatever things that we have covered till now. And we'll be answering the questions that you have. We'll be addressing your needs. We'll be kind of talking about stuff like, what are the things that you can do at your own place. And we will also be talking about various aspects of bioinformatics big data. The time is 3:40 p.m. and it will be in room number 523, Amity University, Newtown, Kolkata, West Bengal. People who are not aware of Newtown, will try to post a direction of that place in the WhatsApp group, or we might also email you. So please get in touch with us if you have any problem reaching there. Please get in touch with us if you have any other problems connected with that. So Chitto, if you are there, if you can say a little bit about this workshop and how do they really reach that place, or maybe you can also talk about the next generation sequencing workshop that you, or next class that you're going to take place on 29th. Chitto?
Thank you, Dr. Pratim. Yeah, the workshop will go on at room number 523 as you've shown. So it is a computer lab, so that each of the participants can actually experience hands-on, so that they can get some essence of this kind of pipeline running by their own actually. So it will be a nice experience for all of them, I think. And to reach Amity, it is very easy to reach Amity actually from the Alia. It is nearby Alia University, and you can take auto from Police Fari, or you can take from Natal Bagan. The auto is available, so it is not that much easy to reach actually. Okay, and for the next class, I think Dr. Pratim has started it very nicely, so I can take the transcriptome part in the next session. So I think you'll be getting a mail containing some files, so that you can run those data in the next session, right, Pratim?
Yes, thank you, Chito. So yes, so Chito Proto will be giving you a lot of exposure about actually how to learn, how to run transcriptomics data analysis pipelines, etc. And we'll also be sending the necessary files which will be there at your desktop. And we will run a lot of pipelines out there on 29th, and of course on 30th, we'll also be try to understand the kind of problems that you're having, because see, since you are running these pipelines for the first time, you'll have a lot of problems. Don't worry about that. We will be helping you over the Skype, over the web, over the email, over WhatsApp, whatever, telephones, whatever is suitable for you. We are definitely going to help you. Thank you, Chitto Proto, for the fantastic, direction. So I guess all of you will be there on 30th and here itself in the Amity. Now, if you can take a few quick questions, because we don't have much time left. So maybe we can end it today and keep all the questions for 30th of July 2019 at Amity itself. So thank you very much. So let's call it a day.
Thank you,
Have a
Pratim.
great weekend ahead. Yes.
Thank
Anybody,
you,
any
Pratim.
question?
Yeah, thank you, Pratim. This is Mohit. Thank you, everyone. I think we had a nice session today, and all thanks to Pratim and Simon. And for the next class, as things have already shown and you guys have already seen. So the next thing would be to go through the genomics and genomics courses. If you have any questions and confusions regarding the pipelines that have been shown today, it's been covered in there. So it would be great if you can just finish that course and go through that course, and you'll have much more clarity. Thank you for everyone for attending today's class, and we'll see you soon on the next class. Thank you.